Hello, everybody. My name is Sylvain Rochon. Nice to see you again this week. I'm here to get people to expand their human experience using cutting-edge technology, as you know. And this is my this week's installment. And pretty excited about this one because this is a little bit like further into the future, but it's today at the same time. And I like these talks, and I like it when it's biochemistry because that's part of my main background. So really interesting stuff. We'll be talking about the road to creating the first human being, and by this I mean actual flesh and blood human beings. Will we be calling them human beings? Probably at first, because we want to start with something we know, and we know our DNA. We actually are starting to understand it pretty well. But first, I wanted to just backtrack before, because it's a road, right? We're not there yet. But right now, what we're working on in the labs is the creation, for example, of human organs for transplantation purposes. So taking a stem cell from your body because you need an organ, and growing a new organ that is still your DNA, and then transplanting the new organ into your body because you have a failing whatever or an accident or something like that. This is something that's where we have very good indicators that we can do that in the next few years. It's in the labs. We've had great successes with animals so far. I have some links below so you can check them out and read about them. So that's very encouraging. And the next step after that, which is actually very similar to creating an organ, is creating an embryo. So without a mother or father, again, taking a stem cell and then creating an embryo that is not inside a womb. So you're actually building an embryo. So you're not touching DNA yet. You're essentially just trying to grow an embryo outside of a womb. And then, well, you probably need a surrogate using this technique. I'm going to again. There's a link below about it, but it's fairly similar to an organ because you're using stem cells to create and manipulate the growth of something based on pre-existing DNA, right? So the embryo, of course, would be a clone of someone in this case, if we're using that topic. But we're able to build embryos, in a new way. So that's better understanding DNA and how things are created from DNA in bits, in parts. Now, one of the challenges about these, in particular the organs, that when you're creating something, you're not creating the blood vessels. There's no heart attached to it, so we have difficulties in bringing oxygen to the cells. And the articles talk about different techniques that have been attempted, and there are more to bring oxygen to the cells as you grow an organ. But I mean, these are experimental first steps. They're very good, excellent ways to improve our health and our medicine. So that's really awesome. But and these are first steps because we're understanding how things go from DNA into something organic, right? And as you probably know, the Human Genome Project, which was like 15, 20 years ago, succeeded in sequencing the first human genome. And we're able to do that much, much faster now. And we've been able to understand and decode in detail about 2% of the genome. Like, we have the full code, but we understand only bits of it. This gene is doing this thing. This gene is doing that thing. I mean, there's like hundreds of thousands of genes in a human DNA. So it'll take a while to figure it out, and hopefully we'll be using AI and computers a lot more to accelerate the process so we can understand it all. And so that's really cool. And in fact, there is a team that is actually using AI, machine learning, computing AI, and these kind of techniques using machines to figure out DNA and be able to make predictions off of it, and for the purpose not necessarily in this case actually creating a new human being so much as understanding it. And in fact, it's probably the path we're going to take if we're able to create a human being, like a full human being, without using the normal processes like in vitro or natural processes and stuff like that, just kind of creating one. It would be a great way to learn how to actually make one. But before we actually get to even that point, we need to reverse engineer the whole thing. So using machine learning, we're creating models, and there's a nice video I found from TED Talks. It's Riccardo Sabatini that talks about his group, that was last year, that have that have coded and built a computer that allows to simulate and analyze and predict just for a sample DNA what a person looked like, age, height, and it allows us to better understand, because it decodes the genetic code itself to do predictions. It allows us to better hone in on different genes and understand what they do, understand what happens, and then you just get like so much better at understanding everything, creating new vaccines, and creating new treatments for diseases, and gene editing and all that stuff. So they're essentially reverse engineering the DNA to understand it, so that we can in fact ultimately create a brand new human being and really get deep into knowledge. I mean, you don't understand. Give you an example. If you've ever built a car engine, if you've never done that, and you look at images, or let's take a new example. Let's take an IKEA piece of furniture. It's simpler. If you never built one, you're looking deep into the materials and the IKEA instructions, and you're going step by step, and you're really looking at the knobs and different things. After you've built the first one and the second and the third, you can you can go at it much faster, and you can be imaginative and decide, well, this plank I don't need, So it's really about building something. Really, when you build something that you can get really good at it. So the road to creating the first human is actually, we're on this road right now with organs, and soon very deep understanding and accelerated understanding of DNA because of Mr. Sabatini's work and his team and others. And then we'll be actually making one in order to even better understand it and be able to heal diseases and all that stuff. So I'm going to let you watch the TED Talks. It's fascinating. It's only part of it. I'm providing you with the link to the whole talk and summary from below. But check it out.
An amazing team, 40 data scientists, and many, many more people. A pleasure to work with. The concept is actually very simple. We're going to use a technology called machine learning. Okay? So on one side, we have genomes, thousands of them. On the other side, we collected the biggest database of human being, phenotypes, 3D scan, NMR, everything you can think of. Inside there, on these two opposite sides, there is the secret of the translation. And in the middle, we build a machine. We build a machine and we train a machine, well, not exactly one machine, many, many machines, to try to understand and translate the genome in a phenotype. What are those letters and what do they do? It's an approach that can be used for everything, but using it in genomics is particularly complicated. And little by little we grew, and we wanted to build different challenges. We start from the beginning, from common traits. Common traits are comfortable because they're common. Everyone has them. So we started to ask our question. Can we predict height? Can we read the books and predict your height? Well, we actually can, with 5 centimeters of precision. BMI is fairly connected to your lifestyle, but we still can. We get in the ballpark, 8 kilograms of precision. Can we predict the eye color? Yeah, we can. 80% of accuracy. Can we predict the skin color? Yeah, we can. 80% of accuracy. Can we predict age? We can. Because apparently the code changes during your life. It gets shorter, you lose pieces, it gets insertions, we read the signals, and we make a model. Now an interesting challenge. Can we predict a human face? This is a little complicated. Because a human face is scattered around millions of these letters, and a human face is not a very well-defined object. So we had to build the entire theory of it. We had to learn and teach a machine what is a face, an embedding, compress it, and if you are comfortable with machine learning, you understand what is the challenge here. Now, after 15 years after we read the first sequence, this October, we started to see some signal. And it was a very emotional moment. So what you see here is a subject coming in our lab. This is a face for us. So we take the real face of a subject, we reduce the complexity, because not everything is in your face. Lots of features and defects and asymmetries are coming from your life. We symmetrize the face, and we run our algorithm. Okay? The results that I show you right now, this is the prediction we have from the blood. Now, wait a second. In these seconds, your eyes are watching left and right, and your brain wants those pictures to be identical. So I ask you to do another exercise, to be honest. Please search for the differences, which are many. The biggest amount of signal comes from gender, then there is age, BMI, the ethnicity component of a human, and scaling up over that signal is much more complicated. But what you see here, even in the differences, let you understand that we are in the right ballpark. Okay? That we are getting closer and is already giving you some emotions. This is another subject that comes in place, and this is a prediction. A little smaller face, we don't get the complete cranial structure, but it still is in the ballpark. This is a subject that comes in our lab, and this is the prediction. So these people have never been seen in the training of the machine. These are the called held out set. Okay? But these are as well people that you will probably never believe. We are publishing everything in a scientific publication, you can read it. But since we are on stage, Chris challenged me, I probably expose myself and try to predict someone that you might recognize. Okay? So in this vial of blood, and believe me, you have no idea what we had to do to have a blood now here. So in this vial of blood, there is the amount of biological information that we need to do a full genome sequence. We just need this about. We run this sequence, and I'm going to do it with you, and we start to layer up all the understanding we have. So in the vial of blood, we predict that is a male, and the subject is a male. We predict that is 1.76 m, the subject is 1.77 m. We predict that is 76, the subject is 82. We predict his age, 38, the subject is 35. Predict his eye color, too dark. We predict his skin color. We are almost there. That's his face. Now, the reveal moment is that the subject is this person. And I did it intentionally. I am a very particular and peculiar ethnicity, Southern European, Italians. They never fit in models. And it's particularly that ethnicity is a complex corner case for our model. But there is another point. So one of the things that we use a lot to recognize people will never be written in the genome. Is our free will. Here's how I look. Not my haircut in this case, but my beard cut. So I'm going to show you, I'm going to just, in this case, transfer it, and this is nothing more than Photoshop, no modeling, the beard on the subject, and immediately we get much, much better in the filling. So why do we do this? We certainly don't do it for predicting the height or taking a beautiful picture out of your blood. We do it because the same technology and the same approach, the machine learning of this code, is helping us to understand how we work, how your body works, how your body ages, how disease generates in your body, how your cancer grows and develop, how drugs work, and if they work on your body. This is a huge challenge. It's a challenge that we share with thousands of other researchers around the world. It's called personalized medicine. It's the ability to move from a statistical approach, where you're a dot in a Gaussian, to a personalized approach, where we read all these books, and we get an understanding of exactly how you are. But it's a particularly complicated challenge. Because of all these books, as today, we just know probably 2%. 4 books of more than 175. And this is not the topic of my talk, because we will learn more. There are the best minds in the world on this topic. The prediction will get better, the model will get more precise. And the more we will learn, every time we will be confronted with decisions that we never had to face before, about life, about death, about parenting. So this conversation, and we are touching the very inner detail on how life works. And it's a revolution that cannot be confined in the domain of science or technology. This must be a global conversation. We must start to think the future we are building as a humanity. We need to interact with creatives, with artists, with philosophers, with politicians. Everyone is involved because it's the future of our species. Without fear, but with the understanding that the decisions that we will take in the next year will change the course of history forever. Thank you.
Pretty awesome, eh? Now the goal is not necessarily to create humans, because we're able to do that right away, like already with in vitro and all these techniques. The idea, like Mr. Sabatini said, is to increase our understanding of it, so that we can essentially manipulate and control. And the first goal of it is really to be able to have healthy lives. So be able to control how long we live. Be able to cure diseases, be able to replace organs, being able to maybe regenerate limbs, things like that. If you understand the genetic code very well, if you're able to build it, you're able to repair it. There's no better repairer of a clock than the person that actually made the friggin' clock, right? So, we're on the path to doing that. I suspect we'll have lab-grown organs within the next 10 years for sure. Maybe even 5. We're going to start being very, very adept with gene editing and eliminating diseases and all that stuff in about 10 years or so. First human being that is synthetic, like made out of our chemicals, like we make the DNA and then kind of make an embryo out of it. I'd figure maybe 15, 20 years from now. This is my guesses because it's really hard to predict timelines in the future. Usually it's faster than we anticipate, and I'm trying to speed it up for myself in here. But those are my predictions. So it's a pretty exciting time because, I mean, who doesn't want to live forever, healthy, young, energetic, and really be able to have full control over how we feel and our comfort levels and our the way we live and how we eat and all that stuff. That's what's coming. So it's really exciting stuff. I'm going to let you go with this. I hope that this gets the noggin kind of turning, and hopefully some of you are into biotechnology and biomedicine. Or technomedicine, and you can find opportunities to actually accelerate all this stuff. That would be awesome. Anyway, that's it for me for now. I hope you're going to have a great weekend and a great week. Ciao for now.